Hello, you are most welcome, ladies and gentlemen, to Real Implantation Moment on Tuesday Night Anatomy with Daniel Okwan. Today's section, we want to begin our journey looking at the general embryology. So we want to look at the activities or the major events that take place in the various weeks of intrauterine life. But for today, we want to begin looking at the main events that will occur in week 1 of embryonic period, and that is mainly fertilization and of course implantation. But because for fertilization to be possible, we need of course the male and female functional gametes by way of course the spermatozoon and of course that of the secondary oocyte being the ovum. I'll also briefly go through the gametogenesis with you. And so, without much ado, let's set the ball rolling. Now, to begin with, one important thing that I want to tell you, regarding these, gametes that we are going to talk about. The gametes that we are talking about, yes, what will happen is that they are stem cells, so to speak, will be coming from what we call the primordial germ cells. So by way of, if it's that of the male, then of course spermatogonium, will be coming from of course the primordial germ cell. If it's that of the female, in the case of female gametes, it will be coming from of course that's the oogonium. It will still be coming from this primordial germ cell. But this primordial germ cell actually arises in the second week of intrauterine life from epiblast. So I'll show you this kind of epiblast when we look at the second week of intrauterine life. But for now, get to know that primordial germ cells, which will give rise to the stem cells of that of the male and female functional gametes, being of course the spermatogonium and of course the oogonium, then they are going to come from this primordial germ cell in the second week of intrauterine life from actually epiblast. Then what happens is that these primordial germ cells, they will actually eventually migrate to of course the wall of the yolk sac. We will look at wall of yolk sac. That is also in the second week of intrauterine life. We look at that. Then, of course, from the yolk sac, around the 5th week of intrauterine life, yes, they will eventually migrate to what we call gonadal ridge. And this gonadal ridge is actually undifferentiated. We don't know whether it's going to become the male or that of the female. But what we are saying is that this gonadal ridge may give rise to either, the primary gonad, or the primary organ. Actually, that's the gonad in female, being the ovary, or it becomes the primary gonad or the primary organ in males, being that of the testes. So simply, what are we saying? The primordial germ cells, which are the stem cells for these gametes to be produced, will be coming from epiblast in the second week of intrauterine life. They will later migrate to the wall of the yolk sac, which is endodermal. We will look at that, and then we will look from there. It will also migrate around the 5th week of intrauterine life or embryonic period. It will get what we call the gonadal ridge. It is this gonadal ridge, which will eventually differentiate to either become the testis in case of the male and that of the ovary in case of the female. So having seen that, we want to look at this process that we are talking about. These gametes therefore will have to be formed. Now in case of the female, what happens is that gametogenesis, and for that matter oogenesis, will happen inside the mother's womb. It will have an in utero commencement, but of course it will get arrested at some point. But for males, it will only happen, gametogenesis will only happen, and for that matter spermatogenesis will happen only at puberty. Yes, at puberty or after puberty, that is where males will go through this kind of gametogenesis we are talking about. And the area where it's going to take place is going to take place in the primary organ of the male reproductive system, and the primary organ is the gonad, and that is the testis. So this is the testis, and within the testis, the important thing that we want to show you here is that we have what we call seminiferous tubules. Now these convoluted structures in the testis, we call it seminiferous tubule. Now those of you who want to really learn about the male and female reproductive systems very well, what happens is that I've done those videos over there, so you can visit my playlist, yes, and then you get all those things. If you visit that of the pelvis and perineum, you are going to get them there. Okay, so but for now, because we are looking at, importantly, the gametogenesis part, and of course continue with fertilization and of course implantation, I will not be going into that much detail. So this one, yes, we are looking at the seminiferous tubules being the site for gametogenesis, okay, in that of males, that is spermatogenesis. Now, what we are saying is that we will have some kind of other processes, but at the end of the day, spermatogenesis, okay, we have certain things, mainly mitotic divisions are going to take place. All these things are going to take place around puberty, with the help of follicle stimulating hormone, interstitial cell stimulating hormone, eventual testosterone and what have you, helping of course Leydig cells to secrete testosterone and what have you. Yes, visit all those videos. Okay, so what are we saying? Yes, this is where spermatogenesis is going to take place inside these seminiferous tubules, and from there they will go through this kind of duct system. Yes, straight duct, go to retidestis, go to efferent ductulis. Then of course we get to this convoluted structure over here on the posterolateral aspect of the testis, and that is what we call the epididymis. What we are saying is that gametogenesis is going to take place over here, spermatogenesis for that matter. Then what happens is that for us to change, to move, okay, I will show you those processes, for us to eventually change from what we call spermatis eventually, so that we get functional spermatozoa, what happens is that they have to be moved into the lumen of this seminiferous tubule, and that is what we call spermiogenesis. Okay, spermiogenesis. Okay, so for us to change it from, of course, spermatis to become functional spermatozoa, that process is called spermiogenesis. That's cyto differentiation process. Then apart from that, these functional spermatozoa may be hanging along where we have what do you call it, the Sertoli cells and all that. They will be attached over there, but they have to be released into the lumen, okay, of these seminiferous tubules, and that process is actually called spermiation. We'll look at that. Okay, that is spermiation rather. But at the end of the day, although it has now become the mature sperm, which we are calling the spermatozoon, it hasn't reached its full maturity. Its maturity will have to also be given, okay, some kind of maturity will take place in what we call the epididymis. Now the epididymis, we have the head, we have the body, we have the tail. It's that therefore at the tail end that we find the functional, let me say the most active spermatozoa. But even with that, they are still not fully mature, and they will get mature in the genital tract of the female. And the main area, yes, although it begins from the cervix all the way, maybe in the uterine tube, but essentially it is going to actually take place in the isthmus of the uterine tube, okay, of the oviduct. That is where mainly, this kind of, gaining that kind of, being very, that process we call capacitation, okay, it becomes very functional. It will take place in the, that of the uterine tube, actually the isthmus of the uterine tube mainly. Okay, so this is, the general bits, okay, that we have to know. So looking at gametogenesis in the seminiferous tubule, the seminiferous tubule is having two cell lineages, and these two cells are the non-spermatogenic series of cells, and those ones, they don't form the stratified layer. Okay, you can see this one is on top of this in that manner. These are stratified. The stratified ones are those ones which form what we call the spermatogenic series of cells. But the non-spermatogenic series of cells, which are just giving some kind of support or some nourishment, and for that matter, we call them supporting cells or sustentacular cells, which eponymously, yes, we call it Sertoli cells. These ones are going to nurse them, nourishment, give them that kind of nourishment, secrete something like androgen binding proteins and what have you. Okay, these are what we call the Sertoli cells. Then apart from that, what they also do is that they form through their what we call processes. They form what we call Sertoli junctional complexes, which form what we call the blood testis barrier. Because what happens is that those ones which, yes, the lumen of the seminiferous tubule, if you are closer to the lumen, you are at the adluminal compartment. Okay, then you are away, you are at the abluminal compartment. So we have the luminal or the basal compartment simply. those are the basal compartment. They are, this kind of what you call diploid, and therefore if the cells of the immune system of the blood and all that gets closer, yes, there wouldn't be any problem so much because they are diploid just like the body cells. But as you move away, going closer to the lumen, then the more haploid cells you are meeting, and those ones, yes, they will have to be taken away from the cells of the immune system. Otherwise, yes, if there's any product that spills into the blood, okay, then there's going to be that kind of autoimmune destruction of these spermatogenic series of cells, which are haploid actually. So please get to know that. But essentially, what are we saying? What we are saying is that from this kind of epiblast, we saw that we had what we call the primordial germ cells. And of course, those primordial germ cells will give rise to this type of spermatogonium. It's actually dark, type A spermatogonium, dark type A spermatogonium. And this dark type A spermatogonium, will undergo, repeated mitotic divisions, depending on the need of, the body. If we need more spermatozoa, it's going to undergo a lot of, mitotic divisions. But in simple, schematically, yes, if this type, A dark undergoes mitosis, then we can give rise to either more of these type A dark, or we give that of type A pale. If you have type A dark, what will happen is that it reserves, it serves as a reserve bank for more of these type A dark spermatogonium to be produced. Then, if you are having the type A pale, it gets committed, so that it goes on with the other processes of the spermatogenic series. Such that what will happen is that this type A pale, will undergo mitotic division, yes, again, to give us type B, spermatogonium. Type B spermatogonium. Now for type B, neither do we have pale nor dark. Go to type B spermatogonium. So it means that through mitotic division, in other words, type A dark, type A pale, both of them are diploid. They will have a 2N, complete set of dark chromosome we are talking about. And then we go to type B. Type B, that's also 2N because through mitotic division. Then also through mitotic division, yes, we also have what we call primary spermatocyte. And this primary spermatocyte, is also diploid. It's also 2N. But it is this primary spermatocyte which will undergo the first meiotic division to give rise to what we call this secondary spermatocyte. This secondary spermatocyte. And this one, yes, becomes haploid. And of course, we know that this kind of, meiosis 1, or the first meiotic division is actually a reductional equation. That is why, yes, primary spermatocyte being diploid, 2N, will eventually become haploid, a secondary spermatocyte, which will have half of the chromosome number you are talking about, N. And that is why, because we know meiosis 1, is the period for, that kind of exchange of genetic materials, between, homologous chromosomes. This is the period where they become that kind of independent assortment of maternal and paternal, chromosomes. So this process, yes, between primary spermatocyte becoming secondary spermatocyte, this area, this stage, is that stage where there's that kind of independent assortment, between that kind of paternal and maternal, chromosomes. So that is what we are saying. Now from secondary spermatocytes, secondary spermatocyte will also undergo, a second meiotic division. Now remember that second meiotic division is an equational division. Therefore, if secondary spermatocyte is haploid, that of the product that we are going to get is also going to be haploid, and that product we are going to get is actually a spermatid. So from this kind of secondary spermatocytes, we get what we call spermatid. Now one thing that we find over there is that spermatids will be joined, through that kind of cytoplasm. Yeah, they will be joined at their points, through some kind of cytoplasmic connections, and that process, where these spermatids, after being formed, yes, losing most of their, cytoplasmic processes, yes, forming that kind of, flagellum, so that it can gain motility, forming that kind of acrosomal cap, so that, yes, those kind of enzymes will get in there. That process, that cytodifferentiation process, so that these spermatids appear rounded, so these ones are actually late spermatids. The early spermatids appear rounded, and then of course, they gain this kind of tail, that's a flagellum, they gain that kind of acrosome, they become elongated. Okay, that one, okay, that process, so that these spermatids eventually become, you know, spermatozoon, okay, is what we call spermiogenesis. So this whole process is spermatogenesis, but that stage, okay, final stage between formation of spermatozoa from spermatids, okay, through cytodifferentiation is what we call spermiogenesis. But what we are saying is that since, yes, so they will still be hanging, yes, around here, but that process, okay, spilling out, okay, moving these functional spermatozoa, okay, into the lumen, okay, so that they can go through, of course, you know, the straight tubules, you know, get to the retitestes, get to the efferent dactylis, and then eventually get to, you know, the ductus epididymis. What we are saying is that that process of spilling them into a lumen is what we call spermiation. So please get to know that. Okay, so this is just a brief description of what we call spermatogenesis. And remember, this spermatogenesis is going to take place, you know, around puberty. And one other thing too that you should know is that what is the period? Usually, yes, some books will say 64, yes, others will say 72. So there's that kind of controversy. But usually, if we are moving from this, usually that date, if you want to get that correct thing, then you have to actually start from that kind of primary spermatocyte. Okay, that one shows that kind of continuous thing that we're talking about. Okay, so in that point, because this type B, after undergoing mitosis, yes, in that order, we may not get the exact age. So usually that date time is calculated from primary spermatocyte onwards, and that one, yes, you get that kind of 64. Okay, but yes, some books also say 72. That's no problem. So the period of, you know, this kind of spermatogenesis, yes, it spans between this kind of 72 and this kind of 64. The reason why we are going to start from the primary spermatocyte again is that this is where meiosis actually starting. And usually when we talk about gametes, mainly it's meiosis we are interested in, not the body wall, which has to do with the mitosis. Okay, so please get to know that. So there we are. This is the early spermatid, which of course you can see that it is, oval or rounded. Yes, having its, you know, nucleus, having the, you know, Golgi apparatus and what have those organelles will be in there. Then essentially, yes, it begins to, of course, undergo cytodifferentiation. Okay, such that, yes, you can see the flagellum emerging out. Then, of course, you know, most of the cytoplasm is shed off. Okay, it's shed, and then eventually it begins to get this, become quite elongated. Okay, the flagellum elongates further. Then there's also that middle piece which you can see clearly with mitochondria, a lot of them also forming in there, because we know that the spermatozoon, functional one, is supposed to be very energetic so that it can actually get focused to fertilize, you know, the secondary oocyte. And then again, at the head end, it bears this acrosome. and this acrosome is rich in, you know, hydrolytic enzymes, yes, mainly hyaluronidase, neuraminidase, yes, these ones which will help, you know, to penetrate, you know, to fertilize that kind of secondary oocyte. So that at the end of the day, the functional gametes we are talking about, although it hasn't reached its full maturity, because, okay, we have to, you know, send it to the epididymis, okay, because in the epididymis, what is going to happen is that, you know, there's going to be this kind of storage over there. Yeah, there's going to be some maturation, and that is why they will gain more motility. Okay, so motility is not really gained, okay, inside the seminiferous tubules or any of these extra testicular duct systems, except that of the epididymis. Okay, so that is mainly what we are going to actually see. Then of course there will also be concentration, and that's why I told you that is the tail of the epididymis that you find a lot of, you know, mature ones, because it has gone through the head, the body, and of course by the time it gets to the tail, yes, it's really, you know, very mature, so that, yes, excess testicular fluid, yes, will also have been reabsorbed. So what are we saying? That process of converting, okay, spermatids to become spermatozoon or spermatozoa is known as spermiogenesis. Is part of, you know, the terminal stage of spermatogenesis. Then these spermatozoon, okay, moved into the lumen of the seminiferous tubule is what we call spermiation. So there we are. this is a section through the testes so that we can see these, seminiferous tube, which are the functional units of the testes. Okay, because that's a factory for sperm production. And we said that we are going to have the Sertoli cells, which are quite elongated. They don't show stratification, and the other ones which show stratification, okay, they are the spermatogenic series of cells. At the end of the day, the process of spermatogenesis, spermiogenesis will take place, will be the terminal stage, converting, converting this kind of spermatids to become what we call spermatozoa, and then spilling these spermatozoa into a lumen. That is what we call spermiation. Well, there may be, there will be connective tissue around. Yes, the connective tissue. Now, so it means that this one is actually the epithelium. Okay, so the connective tissue around, they represent what we call the interstitial cells. So that's the connective tissue interstitial. Interstitial cells, they are the Leydig cells. Okay, and Leydig cells are important in production of, these androgens, male testosterone. Now, on the other hand, for females, okay, we said that for females, then these kind of gonadal ridge that we are talking about, gonadal ridge, will give rise to these primary organs, and they are the female gonads. They are the ovaries. These ovoid structures, okay, find themselves on the lateral aspect of the pelvis. Yes, there's the ovary. Okay, so let's look at what is going to happen. Yes, we want to briefly look at oogenesis. Yes, I've looked at folliculogenesis, oogenesis in some videos that I've done, but briefly for purpose of what we are doing, just a recap. So for oogenesis, yes, we've seen that over there the primordial germ cells will give rise to this oogonium. This oogonium is therefore diploid. Okay, it's with the body, so diploid. This oogonium, okay, now the difference between spermatogenesis, okay, and oogenesis, as I said, is that spermatogenesis is going to take place at puberty, so after puberty, but oogenesis will begin inside, the uterus. And, females usually are endowed with, let's say, about 1 million of these, oocytes, okay, 1 million, let me say, yes, oocytes, because at birth we are going to see what is going to happen, because at birth we have what we call primary oocyte present, okay, but what we are saying is that around puberty, what will happen is that most of these ones will get destroyed through atresia, and then they will be left with just about 400,000, out of which averagely 400, 500 will be ovulated in the woman's throughout the woman's reproductive age, so to speak. So let's begin with this. So we are saying that oogonium through mitotic division, so oogonium is diploid, okay, 2n, through mitotic division, we are going to have what we call this primary oocyte, which is also diploid. Now it is the primary oocyte which then begins, okay, this first meiotic division, meiosis 1. But the interesting thing is that yes, we know that meiosis, we are going to have meiosis 1, meiosis 2. Meiosis 1, yes, the premat, prophase, metaphase, anaphase, and telophase. Okay, then what will happen is that at prophase, okay, prophase 1 of meiosis, actually mainly the diplotin stage, what happens is that this meiotic process is arrested. Okay, it's arrested, such that instead of finally getting a secondary oocyte and the first polar body, we don't get it. It gets arrested, so we don't complete it. It is only going to get completed, at puberty. Because at puberty, what will happen is that, this kind of hypothalamus anterior pituitary gonadal axis will begin working. So what will happen is that there will be this kind of more, LH to be produced, luteinizing hormone will be produced. And when there's that kind of LH surge, okay, LH surgeon, roughly, it surges around, 36 hours, to ovulation. What will happen is that when that LH surge happens, then, okay, around puberty, what will happen is that these kind of primary oocyte will be able to complete the first meiotic division, giving us the secondary oocyte and the first polar body. Well, this first polar body may undergo, further meiosis. That's an equational division. That we said that meiosis 1 is what we call a reductional division. So from 2n primary oocyte, we get n, which is secondary oocyte. Then the secondary oocyte, that's what I'm interested in, because this first polar body may or may not undergo this kind of meiosis. We are not interested in this one, but we go to the secondary oocyte. The secondary oocyte, okay, will then begin this kind of meiosis, 2. And surprisingly, this meiosis 2 is also arrested, but this time around not at prophase but at metaphase. Okay, so please get to know that meiosis 1 got arrested, okay, in the mother's womb, okay, around dipotent state of prophase 1. But this time around, meiosis 2, okay, secondary oocyte, yes, it couldn't complete it. Okay, so it got arrested, okay, at what we call metaphase. It got arrested at what we call metaphase 2. So through ovulation, get ovulated. Now most of the oocytes in females do not get complete this second meiotic division. Why? Because they don't get fertilized. The only time the meiosis 2 gets completed, okay, is when it gets fertilized by a functional spermatozoon. Okay, so that is when we are able to complete the second meiotic division. So that, yes, we know that second meiotic division is an equational division. So from haploid secondary oocyte, we are able to get this kind of ovum. Okay, yes, ovum is still secondary oocyte anyway, and then we get our second polar body. So that is what we are talking about. And of course, you know that the zygote which will be formed will therefore be diploid, because we have an N coming from the functional spermatozoon, then we have an N from the ovum, okay, forming this kind of so that is what we have to know. So there we are. Now one thing is that, yes, there will be several spermatozoon, but actually just one of them is able to penetrate, okay, and then fertilize this kind of secondary oocyte, being the ovum. So that is that image showing you. So there we are. Now, so look at this. This sperm, for it to be able to fertilize this ovum, then it has to go through some kind of barriers. And for it to do that, then it should be very energetic. It has this flagellum so that it can swim. The energy supplied from this mitochondria. Then, of course, the acrosome, okay, at the anterior aspect of the headpiece, releasing this kind of enzymes to break through it. So it has to break through this corona radiator cells. It has to also go through this kind of zona pellucida. Okay, we also have that of what we call vitelline membrane. It has to go through, then it goes through what we call the oocyte membrane, the plasma membrane of the oocyte itself, because before it can get into the what we call the cytoplasm of the oocyte, and for you to fertilize with it. Okay, so that is what I'm talking about. Now when they get fertilized, what happens is that the key thing before fertilization, when the head of the oocyte enters, we form what we call the pronuclei. Okay, so we have the male and of course the female pronuclei, okay, forming. So we can see they are separate. So one of them is the male and the other is the female pronuclei, okay, within the cytoplasm. Then what happens is that then these pronuclei will fuse, okay, they will come together and then fuse. So that's the fusion of this pronuclei we are talking about. Okay, so it's not just a single event that there's that fusion. There's first formation of these kind of pronuclei, then these pronuclei will have to fuse. So as I said, there are millions of spermatozoa that are released per ejaculate. That 2 to about, let's say, 3 mil of ejaculate, okay, will have about 200 to about 300 million this kind of spermatozoa or sperm. And what is happening is that, yes, average 250 million. Average, what happens is that the female reproductive tract, yes, the vagina, yes, is quite acidic and therefore hostile to these sperm. So what is happening is that most of them, okay, will get sacrificed. Yes, most of them too are not really morphologically agile to fertilize that of the secondary oocyte. Therefore they will die on their way or something like that. So what is happening is that roughly 300 to 500 sperm are able to reach, okay, the area of the secondary oocyte. However, only one is able to, fertilize it. So there's that kind of great competition over there. And I told you of the barriers. Yes, we have these granulosa cells over here forming this kind of corona radiata. How to penetrate it? Yes, you also have to go through this kind of zona pellucida, that acellular belt of glycoprotein over there. Yes, you have to penetrate it. Then you also have to go through that kind of vitelline membrane to get eventually to the membrane of the oocyte. But over here in that region, spanning around the membrane of the oocyte, okay, that of the vitelline membrane and that of the glycoprotein, zona pellucida, are receptors. Okay, so we call them sperm, receptors. Okay, so sperm receptors are over here. Okay, they span over here all the way. So what is happening is that the spermatozoa are supposed to bind, okay, cell surface receptors, which are over here, before that kind of they can fuse with it. Okay, so once we're able to go through that, then what will happen is that we will have to prevent what we call polyspermy. If one, sperm is able to fertilize it, then we have to prevent other sperm from actually penetrating. So what are we saying? We have a lot of spermatozoa, okay, reaching that area, about 300 to about 500 of them, okay, and they're able to, secrete that that enzymes, okay, in the acrosome. There's that kind of acrosomal reaction, okay, so that they can secrete that kind of enzymes. Okay, so if I talk about enzymes by way of the hyaluronidase, neuraminidase, yes, you have to penetrate it. Okay, so once they're able to do that, then they're also able to break through, okay, or interact with the zona pellucida, because there are also some receptors. Okay, zona pellucida, there are some proteins, protein 1, protein 2, protein 3. Yes, they are also present, so it has to interact with it. Okay, so that's what we call the acrosomal reaction, that enzymes release to digest, okay, this kind of corona radiata, zona pellucida. Okay, then the vitelline membrane. Now the key thing over here is that one sperm is able to succeed, okay, to go through all these barriers we are talking about, eventually penetrating the plasma membrane, okay, or the cell membrane or oocyte. Okay, so that is an interesting thing. And so the question is, how are we able to prevent other sperm from fusing, okay, or from also fertilizing the oocyte? And that is what we call prevention of what we call polyspermy. And in the prevention of polyspermy, there is something we call cortical reaction. Okay, so at the cortex something happens, the outer aspect something happens. And what are we saying? What we are saying is that I told you that there are some receptors over here, and those receptors, yes, they span from this kind of vitelline membrane, yeah, zona pellucida, and that of the membrane of the oocyte. Okay, so that's the sperm cell receptors, and these receptors, that's where these sperm are going to bind. Okay, so what are we saying? What we are saying is that between what we call the membrane of the oocyte itself and that of the vitelline membrane, okay, we have these granules. Okay, we have these granules. Okay, these granules are over here. Now, what is going to happen is that the moment one sperm is able to penetrate the membrane of the oocyte itself, okay, then what happens is that these kind of granules will go through a process, and that process we call cortical reaction. And these granules will ensure, okay, that these receptors which the sperm is supposed to bind to, they begin to get removed. Okay, so these sperm cell receptors, okay, on the surface which span over here, get removed. And then again, the vitelline membrane also gets hardened, and then an envelope forms, and that envelope is what we call fertilization envelope. Okay, and this one is able to block other sperm from actually penetrating or fertilizing the oocyte. So only one is able to succeed because of the process we call cortical reaction. So now, once there's that, we formed it. Yes, we said that the male and female pronuclei, yes, eventually will come together, will fuse, okay, and then they get fertilized. Once we have this, then what happens is that this, one cell zygote which we formed, okay, one cell zygote which we formed, yes, will undergo division, mitotic division. That's what we call cleavage. And this cleavage, if it's one cell, when it goes cleavage, we get 2 cell stage. But the interesting thing is that the cleavage which is taking place, it is, let me say, asymmetrical. Okay, it's not entirely the same. So what happens is that one will be larger than the other. Okay, the two cell stage, one will be larger than the other. So it means the larger one will therefore undergo the next, cleavage. Okay, so for instance, assuming this is the larger one and there's a smaller one, this one will divide further so that we get the 3 cell stage. Then because this one, yes, now this one has become the larger one, it will divide so that we get, of course, the 4 cell stage in that manner. So we will go through that. Yes, so roughly the 2 cell stage happens around 18 to about 36 hours. Okay, now one thing that I want to tell you is that fertilization, okay, usually, yes, between ovulation and fertilization, usually the difference is just about 24-hour interval. And then we go through, of course, the 4 cell stage, and in that manner eventually having the 8 cell stage, around 48 to about 60-hour period, about 48 to about 60-hour period. So that is what we are able to see. Now one thing that we find is that this kind of cleavage, it goes on and on until we are having the 8 to 16, 8 or 16 up to about 32 cell stage. Roughly, sometimes we use 32. So it's 8, even 16 or 32. But the key thing is that it's forming a ball-like structure. Okay, that ball-like structure is what we call morula. So this one is the morula. Now the interesting thing we find is that, yes, whether it's a 2 cell stage, 4 cell, 8 cell, whatever cell stage, we have this thick glycoprotein that I talked about, which of course we said that the functional spermatozoon will have to penetrate it. Okay, still it is present, and it is present because it will prevent premature hatching of the zygote. Because when this glycoprotein hatches out, then what will happen is that this is the important thing, it will get implanted. And if it implants at a wrong area, then there's going to be that kind of ectopic pregnancy. So that, yes, this kind of fertilization is going to take place inside, okay, inside the uterine tube, mainly, yes, in the ampler region. But some should also say amplo-isthmic junction, yes, but mainly ampler region. Yes, that is where fertilization is going to take place. So if it takes place over there and there's that premature, okay, breaking or disintegration of the zona pellucida, yes, then implantation is going to take place there, and there's going to be that kind of tubal ligation. And of course, what may happen, yes, rupture of this oviduct, of this uterine tube we are talking about. Okay, so that is what you have to actually know over here. So what are we saying? Yes, at every month in a reproductive, active female, okay, a woman beyond puberty but not up to menopause, okay, what is happening is that, yes, the ovary, okay, contralaterally every month, what will happen is that one ovary will release one oocyte. Will release one secondary oocyte, and that process is what we call ovulation. And the ovulation, yes, we know that that secondary oocyte is arrested. Okay, its second meiotic division is arrested at metaphase 2, and then through, yes, one sperm will fertilize it so that we form the male and female pronuclei. Then eventually these ones will fuse, okay, so that we get the zygote. And that zygote we have, yes, immediately it almost always starts dividing. Okay, so it starts undergoing that kind of division, and that division is what we are calling cleavage. Okay, so it goes through the 2 cell stage, 3, 4, in that order. But in the uterine tube, actually starts somewhere in the cervix, even in the uterine tube, in the fallopian tube, wherever. Okay, what is happening is that the sperm, okay, they will have to get what we call it, get the fully mature. Say that glycoproteins, okay, which are covering the zona pellucida, which are covering the acrosome, yeah, they all have to be removed, okay, and they become very functional so that they can fertilize this secondary oocyte. That process is what we call capacitation, and it's mainly going to take place around the isthmic junction. So the isthmus of what we call the uterine tube, that's mainly where it's going to take place. Okay, so that is that one. So several cleavage, and compaction will take place. So what is happening is that the cells, they actually, get compacted, and that's why they are able to form that kind of ball-like structure, which we call the morula. Now, one thing that we find is that once we have this kind of morula, okay, once we have this morula, which of course we said is the 8 cell stage, or even 16, or even up to 32 cell stage, what we are going to see is that this morula, actually the late morula, and whenever we say the late morula, look at these cells. These cells are more or less together. They've compacted together. What will happen is that the cells get differentiated, so that some of them will form the outer rim, and then some of them will get compacted at one end, okay, and that those one we get at one end, okay, is what we call the inner cell mass. Over here, inner cell mass, and those ones at the periphery we call it outer cell mass. It is still morula, but that's the late morula stage, and usually it's around day 5 that is able to enter. Day 4, day 5, mainly day 5 is able to enter the uterine cavity. Then around day 6, okay, it begins to get implanted. Now the question is, where is this implantation actually going to take place? Now at the point of implantation, we no longer call it morula, late morula, because uterine fluid will seep into that cavity over here, that region between them, okay, that space over here. Uterine fluid will enter, so that this area becomes the blastocyst, okay, blastocyst, because over here, this now, once the fluid has entered, called blastocyst, we now call this blastocyst. So we are having the zygote, we went to 2 cell stage, 4 cell stage, whatever, up to 8, 16, or the morula 32 stage, whatever. Then it organizes itself before it entered, okay, it organizes itself into an inner cell mass and outer cell mass, okay, before entering the uterine cavity. It's a late morula. Then once it enters, fluid from the uterus seeps into that cavity between the inner cell mass and the outer cell mass, okay, that cavity is called blastocyst. And then what is happening is that structure, okay, that structure is that structure for implantation. That structure is what we call, blastocyst. And we say that implantation, it roughly begins around day 6. By the end of the day, it got completed fully, okay, by day 10. So by day 10, you should have gotten completely burrowed into the uterine, wall. Now the interesting thing is that some people think that yes, that implantation is just attaching to the wall. No, it gets buried. It goes deeper, okay, it goes deeper into it. And that is why in human beings we describe it as having that kind of interstitial implantation, because there's that kind of burrowing into, of course, the uterine cavity, the wall actually. And we will see some people called decidua, okay, it gets burrowed into it, okay. So that is what we have to actually know. Now, where is this implantation actually taking place? It's taking place at the posterior aspect of the fundus, okay, mainly. But sometimes too, you may find in some books that they will get implanted at the junction between the posterior aspect of the fundus and the upper aspect of the body. Okay, that one too is also correct. But mainly posterior aspect of the fundus of the uterus. That's where implantation will take place. If it takes place yet at a lower level, as it comes more down, then of course placenta previa is what we call intrauterine ectopic pregnancy. Okay, that one we don't like it. We want the normal one, which is going to be the posterior aspect of the fundus of the uterus. Now again, sometimes if you want to extract, okay, or if you want to harvest embryonic stem cells, yes, usually we get them from this blastocyst stage. Okay, this is where you get this kind of embryonic stem cells. Okay, so please get to know that. So there we are. That's the blastocyst I'm talking about. Okay, there's the external view. So if you cut through it, then you're able to see the internal aspects. Okay, so those ones which form the outer aspects. Okay, that's what we call the outer cell mass. And the inner portion, we are going to call it inner cell mass. And that space is what we call blastocyst. So this is what we call blastocyst. That is the implantation structure. So it's not just the zygote or the 2 cell stage or the morula, but it is the blastocyst. So once we are through with implantation, that is the end of what we call the first week of intrauterine life inside the womb. And that one, yes, what will happen is that the uterus is having 3 parts. We have the perimetrium, outermost, we have the myometrium, the muscle coat, we have the endometrium, okay, the inner coat. So what is happening is that the conceptus, okay, or the blastocyst, it actually burrows into it, okay, so it burrows, it goes into it. Now that portion, once, after conception, what will happen is that the endometrium is now given a different name, and that's what we call decidua. Okay, so if you hear the word decidua, that is, simply the endometrium, okay, of the womb, but this time after implantation, okay. And this decidua, okay, that region, okay, which is over now, there's a conceptus, that region below, okay, or that is under or inferior to, of course, the conceptus, okay, the endometrium below it is what we call decidua basalis, okay, so base, decidua basalis. Then we have that decidua, which of course is actually between the lumen of the uterine cavity, okay, as well as the conceptus, okay, or the site of implantation. This one is what we call decidua capsularis. And then you also have under decidia, that decidia, okay, which form part of the wall of the uterine cavity, which the implantation didn't occur there. The remaining portion is what we call decidua parietalis. Of course, you know the word parietal has to do with wall, okay. So that is the main thing we are talking about. So please, for this section, we're able to look at gametogenesis, talking about spermatogenesis, and of course oogenesis. Then we also actually looked at what is mainly going to happen at week 1, that is fertilization and implantation. So ladies and gentlemen, I hope you find this video helpful. If you've not subscribed to the channel, please do, so that yes, in future videos like these ones, you will be able to enjoy them. I'm very grateful for your time this evening. Have a good night, and may God richly bless you.